Minasan, ohayo gozaimasu. Buenos días a todo el mundo. Buenos días. Kyou wa kinyoubi desu kara. Omedetou. Shumatsu wa chikai desu kara, ne, omedetou. Enhorabuena, enhorabuena porque hoy es viernes y como siempre os digo, como es la entrada al fin de semana, pues enhorabuena y felicidades y que aprovechéis bien el fin de semana. Vale, y ahora llegamos ya al momento de la presentación. Quiero presentaros a Pierdo. Pierdo es, bueno, habéis podido leer en la biografía que os pasé y tal. Bueno, pues Pierdo, aparte de que nos conociéramos ahí en Japón, yo estaba haciendo la tesis doctoral cuando él llegó a hacer un postdoctorado. Con posterioridad volvió a Italia, pero desde entonces ha tenido una trayectoria científica absolutamente espectacular, extraordinaria, con muchos éxitos y a lo largo de, además de tener una gran carrera científica, la ha tenido también desde el punto de vista, digamos, de gestión de la ciencia política. Dado que él fue durante 6 años, fue el rector de la Escuela Superior de Santa Ana. Esta universidad pública es una de las 10, está catalogada como una de las mejores 10 universidades públicas jóvenes de Europa, de menos de 50 años. Tiene unos ratios de éxito espectaculares, reclutan a los 1000 mejores estudiantes de Italia, aquellos que son reclutados, pues los becan, pero a full. O sea, desde los estudios, alojamiento, todo. Y son la base de, en cierto modo, de la economía política y tal en Italia. Por ahí han pasado varios primeros ministros, ¿no? Comentabas ayer. Bien, pues tenemos el privilegio de tener a Pierdo. El otro día, hablando de este tema, tiene en su currículum tiene un positivo, más allá de lo que es la ciencia, también la gestión. En su exitómetro hay que destacar un parámetro, que es el hecho de que él me decía, cuando me fui tenía el mismo número de enemigos que cuando entré a dirigir la universidad, la Escuela Superior de Santa Ana. Pierdo desde el punto de vista científico, ya digo que es extraordinario. Gran parte, o si no la totalidad de su vida científica ha estado dedicada a investigar la hipoxia en plantas. Ya cuando lo conocía y en Japón era un tema que él trataba. Y luego, pues, temas de señalización de azúcares, también es una persona de referencia en el mundo de la ciencia en plantas. Y luego, pues, en los últimos años también ha estado muy volcado con el tema de los small RNAs y microRNAs y todo esto, ¿no? Y entonces, bueno, pues ya ha llegado el momento de cederle la palabra a Pierdo y que nos deleite con su exposición, con su charla. Sí. Oye, un aplauso a Pierdo.
No te preocupes. Ya está. Okay, so I never had such an introduction in all my life, so it's kind of difficult to start after all this, and especially after seeing my photos of myself back 33, 34 years, probably. So it's great to be here. I had a great time in these days here in Malaga and visiting your institute and talking with Javier and many of you in these days. But now it's time for me to make my part of the job and therefore presenting my seminar. So the topic as introduced by Javier is hypoxia, and I've been working on this since my master's thesis. So, I must say that, so it's more than 40 years that I work on this topic. I must say that during the first 20 years, I kind of regretted of having chosen that topic, because nothing really exciting happened during the first 20 years. But then, everything happened. So I'm glad I didn't give up during the first 20 years. So this is also for younger people. You might think that the topic of your work is not that exciting. It might be the case, but it might also not be the case. And so it's worth trying and seeing whether something really new might happen in your field. And this is, so most of the things you will see in my presentation happened during the last 20 years. So I will not go back 40 years about that. So what is hypoxia, and why am I talking about the many facets of hypoxia? Because during the last 20 years, we realized that hypoxia is not only too much water, but is more than that. So I will go briefly talking about this part, but then we will move to something quite unexpected that brought hypoxia to be a more important scientific topic in plant biology. So hypoxia is actually the lack of oxygen, and in the environment it happens when it rains too much. So it's straightforward. Too much water. These plants here do not have access to oxygen, and since they need oxygen for respiration, they will eventually die. It doesn't, they do not last longer than a few days under this condition, then they are gone. And this is kind of a problem. So I changed the slide to include some very recent data. So this is the precipitation rank during February and March, and you can see that you know that. So okay, so and it's quite impressive how rain is becoming a problem, despite of the fact that over the last decades we were told that drought was gonna be the real problem, which actually is, but is just one of the two aspects of the climate change. On one side, there will be little water, drought, but on the other side, this might happen, so too much water, and this has an impact on plants, not only on the infrastructures, but also on the life of plants, and therefore on agriculture. So when I started working on hypoxia, the scientific question was, how do plants survive under hypoxia? Or better to say, how can we make plants more tolerant to hypoxia so that we can have plants that are able to grow under partially flooded soil, etc., etc. And the model system we took was rice. Because if you want to study a crop, why rice? It's 40 years ago, Arabidopsis was not known. So it's not like today that you want to tackle any scientific question, you start with Arabidopsis, and then eventually you move to a crop. Here we started with a crop, and you will see eventually we move to Arabidopsis. So we started with rice because rice is a perfect model, okay? It clearly can survive under conditions that cannot be tolerated by any other crop. So it's the perfect crop to be studied in order to understand how it survives under flooding. And as I said, nothing happened for 20 years. So yeah, we'll try and try and try. There were pieces of a puzzle accumulating, but the picture was unclear. At a certain point, everything came clear, and we understood, not only us, I mean, it's part of the results of a scientific community, especially scientists in the Philippines, who and in Japan, who using genetics unveiled the mechanism of tolerance for rice. So I will start with the mechanism of tolerance of rice, and then we will move forward to see other things. So How is rice able to tolerate flooding? Well, it is able to tolerate flooding, especially when the water level increases a lot, by keeping the leaves above water. Okay, so this is how this farmer is surviving. It's a real photo, it's not my... He's not hypoxic. He would be hypoxic if he couldn't breathe, but since he's keeping his head above water, he can breathe because he can breathe, and then oxygen will be translocated to the underwater part. And these plants of rice do exactly the same. They simply have the leaves above the water level, and they have a very efficient transport system of oxygen, so that oxygen can be translocated to the parts of the plant which are submerged. But how can these plants be able to keep their leaves above the water surface? Well, there are a variety of rice which are able to grow in Asia, which can grow up to 3 meters tall, because the water level can increase up to 3 meters. And how do they do that? Well, they do it very rapidly. This is a plant, a control plant, and this is a plant in which we raised the water level in 6 days. And you can see that it grew up in 6 days almost twice the initial height. So it's a growth rate of 30 centimeters a day. It's quite incredible. And how does this plant do that? It does that because when the plant is submerged, ethylene accumulates, because it cannot, ethylene is a gas. If a plant is in air, it will go away. If the plant is submerged, it will stay in the plant. The level, the concentration of ethylene in the plant will increase, and 2 genes called snorkel genes, obviously, are activated, and they activate the growth triggered by gibberellins. So it's really straightforward, simple. Ethylene accumulation, induction of the snorkel genes, activation of gibberellin growth, and the plants start growing very fast. So these 2 genes belong to a category of transcription factors. They are called ERF group 7. ERF stands for ethylene responsive factor. Group 7 is because it's a very large family of transcription factor, and these are group 7. I'm mentioning this because this will lead to the inverse translational biology from rice, from a crop to a model plant. Okay, so it's a ERF. So if you think of it, you can have rice cultivation here in Spain having this behavior. Forget it. This is a behavior that only a certain group of varieties, that are called the deep water varieties, are able to do. Okay. The rice we grow in Italy, the rice you grow in Spain, do not have snorkel genes. Because this is one of the many adaptations of rice varieties to submergence. Other rice varieties adopt a completely different strategy. Again, it's not the rice we cultivate. The rice we cultivate in Europe is dumb. Okay, it actually can survive very shallow submergence. So if it is completely submerged, it will die. But there is another group of varieties which are cultivated in Asia, which when completely submerged, they adopt a completely different strategy. They do not start growing fast, but instead they decide to enter into quiescence. They do not grow. Okay, which is the mechanism that our varieties do. Our varieties try to grow a little bit, but not fast enough to keep the leaves above the water surface. They try to escape submergence, but they are inefficient. They fail, and by trying to grow, they use all the sugar reserves, starch, and nothing is left for regrowth when the water level decreases. So our rice varieties are not very good, but these varieties are very good because they adopt another strategy, which is a quiescence strategy, so that when the water level decreases, growth can restart. Okay? This is possible, again, because ethylene, which accumulates, it induces another gene, which is submergence 1, which activates repression of growth. So it's exactly the opposite strategy as used by the deep water rice varieties, in which ethylene induces a very fast growth. Here, the ethylene via this transcription factor activates repressors of gibberellin growth, and this stops growth of a plant. The plant does not grow at all. It waits until the water level recedes, and then the plant restarts. This mechanism is more interesting also for us, because having plants that are 3 meters tall is not agronomically interesting, because you can imagine when water recedes, these plants are not very stable. But in this case it is, and these are the results of improving rice varieties with the sub-1A gene, and these are improved varieties called scuba rice. Again, the names are evocative of the mechanism, and you can see comparing the traditional rice varieties like the one we grow here in Europe. What is left after a submergence event? Most of the plants are dead. These rice varieties, the scuba rice varieties, are very tolerant, and they will lead, and they are already leading, because the commercial varieties are already in place for this with this trait. They will lead to food production, sustainability of food production in Asia under the threat of climate change, so increased flooding events. So again, this gene belongs to the ERF7 group of transcription factor. So when all this came out, we are around, so it's less than 20 years ago, so more or less. So everyone was quite excited. Finally, we know why rice is tolerant. And the obvious question was, are these genes important in all plants? Are they doing something which is possibly explaining the mechanism of adaptation of plants to low oxygen? And therefore, we started to investigate from rice to Arabidopsis, which is the unusual shift from a crop to a model plant. So it's quite funny that in this case the breakthrough discovery came from a crop, from research in a crop, and then was translated to Arabidopsis. So this is a photograph we made in my lab. It's simply an Arabidopsis plant which is kept underwater. Of course, the plant will eventually die. Okay, so it's not really well. All plants have different degrees of tolerance. Okay, it's not very tolerant. It can tolerate a few days, but then it will die. So we started working with Arabidopsis, and I will cut a very long story short. It was quite complicated. So in the beginning, we didn't understand really much of what was going on. But the key experiment, I would say, was a confocal imaging of these ERF proteins. Because, of course, there are ERF proteins in all plants. Okay, so the ERF7, ERF ethylene responsive factor, group 7, are present in every plant. They are in tomato, I'm sure they are in mango, in papaya, in avocado, and etc. So it's a family which is very well conserved. So what I'm going to tell you is reasonably valid in any crop. So it was not really clear by doing the classical gene expression experiments, overexpression, knockout, not really clear what was going on. But when we looked at the protein at the confocal, this is what we have seen in air. So you see that the GFP, which is the protein, is in the plasma membrane. So the gene is always expressed. But if we move the plants to hypoxia, the protein goes to the nuclei. And if we reoxygenate, it's gone. So the hypothesis here, without understanding yet what was going on, was that somehow the protein is oxygen unstable. So under hypoxia, the protein is where it was supposed to be. It's a transcription factor. You expect it to be in the nuclei. If you reoxygenate the plant, it's degraded. Somehow it disappears. So it takes some time before going back, because eventually from here, you will get back to here. But then it means that the protein, which is likely to be oxygen unstable, gets stabilized by docking to the plasma membrane, which is exactly what happens. So the protein is an oxygen unstable protein, which is stabilized by docking to the plasma membrane, and we know also to which protein it docks to. But it is eventually released from the plasma membrane, migrates to the nuclei, where it activates the transcriptional activation of many genes which are required for the plant to tolerate low oxygen. Okay? So it's a master gene which activates the response to hypoxia, and the master gene is apparently an oxygen sensor. Because a protein which is stable under hypoxia and unstable under oxygen, it means that it works like a switch on-off, okay? And oxygen is the trigger for switching on-off. Oh, better. On when there is no oxygen, off when there is oxygen. So how, again, to cut along, that work was published very well. Both my group and another group, we published together in the very same issue of Nature because of these weird things that sometimes happen. Two independent groups reached independently the same conclusion, and we succeeded in avoiding to have one group killing the other, okay? So it's weird. It was worrying at the time. Now I'm laughing, but I was not laughing at all. When we discovered that there was a competing group and we were 1 month to submission, it was not funny. Okay, so, but everything was fine. We published it. And what did we discover? Well, we discovered that this is the name of the ERF7 protein, okay? One of the 5 which are in Arabidopsis. This is probably the most important. So this is an ERF7 protein, and it has one characteristic, which is the key of the mechanism, is that it has, of course, a methionine at the N-terminus, followed by a cysteine. The methionine is removed enzymatically, and the cysteine is exposed. And here is where the oxygen-dependent mechanism occurs. If a protein, not just this one, theoretically any protein having this N-terminus, an MC at the N-terminus, is oxygen labile. It is I anticipate that is not always the case, so it's not sufficient. Having an MC is not sufficient, but in order to have a protein which is oxygen labile, you need to have an MC at the N-terminus. With oxygen, the cysteine gets oxidized. The protein with the oxidized cysteine gets arginylated and eventually ubiquitinated. So it's tagged for destruction. So a cysteine is no problem. Having a cysteine here is no problem. But if a cysteine gets oxidized, then it is targeted for degradation. This is called the N-degron pathway, where what is at the N-terminus of a protein defines the fate of a protein. Okay? If we had an MA, it would be very stable in the presence of oxygen. But since it has a cysteine, it is unstable. So this is the mechanism. So when there is no oxygen, the transcription factor is stable and active. When there is oxygen, the transcription factor is degraded. And this is the oxygen sensing mechanism. So oxygen is the key. And under hypoxia, oxygen is absent. And we have the transcription factor going to the nuclei, activating transcription, having anaerobic proteins, and eventually having some kind of tolerance, okay? Not as much as in rice, but it is actually essential for having some tolerance in Arabidopsis as well. So how does cysteine oxidation occur? And this, it took an extra 3 years to reach this result. Because there were two hypotheses. One is spontaneous oxidation. There is oxygen, there is a cysteine, it will oxidize. Oxygen will oxidize it, or it is enzymatic. It is enzymatic, and we discovered that this is the reaction. So cysteine is converted to cysteine sulfinic acid. So this is the chemistry of the oxidation of a cysteine at the N-terminus. As I said, it could have been oxygen. Nitric oxide is somehow involved, not directly in the reaction, probably downstream. and oxygen is required for this reaction, which could have been non-enzymatic, but it's actually enzymatic because Dan Weitz, who was a postdoc in my lab, when he was working as a PhD student in Germany, he found, together with Francesco Licausi, that who was in my lab, that PCO, which is the name we gave to the enzyme, which is plant cysteine oxidase, is a cysteine oxidase. Cysteine oxidases are enzymes that oxidize cysteine, but this is requiring that the cysteine is part of a protein. So it's an enzyme which is a cysteine oxidase, but it does not oxidize the free cysteine, but it only oxidizes cysteine which is at the N-terminus of a protein. And this enzyme is the one which oxidizes cysteine to cysteine sulfinic acid, targeting the protein for the destruction. So this is the mechanism. You have MC proteins. There are, in Arabidopsis, there are around 200 proteins. So in the Arabidopsis genome, there are 200 proteins which have an MC. Okay? Statistically, this is telling us that the frequency of having a cysteine at the N-terminus is lower than that expected by chance. Okay? So it suggests that having a cysteine, and actually this is the case, so having a cysteine there is tricky for a protein because it will be regulated post-translationally by the presence of oxygen. And oxygen together with PCO oxidize the proteins which are then degraded. So this is the mechanism summarized. So we have ERF7 protein, we have a cysteine, PCO oxidizes the cysteine, and they are targeted to the endegron pathway and degraded when oxygen is present. It's very interesting that this is very similar to what happens in mammals, in us, because we do have an oxygen-sensitive mechanism, which is that one. Different protein, different mechanism, but very similar. We have a protein which is called the HIF1, which has prolines, which are oxidized by an enzyme, which is a PHD, which is a proline oxidase. So proline gets oxidized and eventually is degraded. So this is interesting because it's telling us that evolution did kind of the same work in plants and animals, exploiting different mechanisms for oxygen stability and stability in the organism. And quite interestingly, after we discovered this system and realizing that it was similar to the human, when in humans, we found that in humans also there are cysteine proteins, and there is an enzyme which was discovered, which is analogous to the plant 1, which does exactly the same. So there is a second oxygen sensing mechanism in humans, which is borrowed, let's say, from plants. So this is quite interesting because, as I was telling you, the story is we started with rice, then we moved to Arabidopsis, and eventually we ended up with human beings. So it's a nice story, and that nice story was told by Peter Ratcliffe, which we collaborated with us, and he got the Nobel Prize for the discovery of oxygen sensing. And it was one of the first times that in a Nobel Prize lecture, the awardee included one slide with a plant. Because, no, I mean, it's important, because you know what plant blindness is. We work on plants, but regretfully, plants are not seen by people as important. We know they are important. We go to the supermarket every day and we buy plants and we eat plants. But even in the scientific field, we are perceived as, okay, you work on plants. I am a biologist. What do you work on? Plants. So it's not that exciting. But this is telling you that it is possible to make important discoveries in plants that actually can translate to, we can go back and forth from plants to animals, and this is also, again, for young people. Always, when you are studying something, try to have a look to the literature in the animal field, because there is more money in the field and more people, they are going faster. So it might be interesting to try to understand whether there is any interesting hypothesis in the animal field which can be borrowed and brought to your field to discover something new. Because plants and animals are much closer to each other than we tend to believe. Okay? So that was it. So, okay, done. Finally, we can change topic. But we started thinking: the idea is why did evolution fix such a complicated mechanism to sense oxygen just for a few days of extra rain? So, are we sure that all this oxygen sensing mechanism was evolved because of rainy days? Because we find this mechanism. If we had found this mechanism to be specific to rice, specific to water plants, but it's present in plants which grow in the desert. So evolution did not select this mechanism because it was useful when it rains too much. That was the idea. So there should be, hypoxia should be part of the life of every plant every day in order for the plant to need a mechanism to measure oxygen, okay? So we started thinking about hypoxia without flooding. And actually, hypoxia without flooding exists. This is what happens. This is the blue color is a gas staining of an hypoxia responsive gene. And you can see when you infect an Arabidopsis plant with botrytis, at the infection site, you get activation. And actually, the oxygen level in a leaf of Arabidopsis, which is infected by botrytis, drops down to less than 1%. So for any time a plant is infected by a fungi, hypoxia is there. But not only hypoxia is a consequence not only of abiotic stress like flooding or biotic stress like a pathogen infection, it is also present in apple fruits. So you work a lot with fruits. Be sure that inside an avocado, inside a mango, inside a papaya, there is no oxygen. This is an apple fruit, and you can see that the oxygen can be close to 20% outside. It may drop to close to 10% or lower in the inside. The reason is obvious. Diffusion of oxygen in liquids is low. So if the tissue is a thick tissue with no air spaces, there are very little air spaces in an apple, diffusion of oxygen is limiting the presence of oxygen inside. The same applies to a potato, okay? Any bulky organs are hypoxic inside. And inside an apple or inside a tomato or inside, I would expand this to any fruit, hypoxia is sensed through the ERF7 mechanism, which I just explained to you, and it activates the classical metabolic adaptation, which uses efficient utilization of sugars through glycolysis to produce ATP, compensating the lack of ATP through aerobic respiration, because if there is little oxygen, the mitochondria will not be very efficient. So you need to compensate for the lack of ATP not coming from mitochondria, with ATP coming from glycolysis, and with ethanol as the end product. This is the classic. When I started my thesis, that was what was known about hypoxia. Plants under hypoxia produce ethanol, which is actually the case. So, which are the consequences of having ethanol in the inside of an apple fruit? Of course, the apple is doing that to adaptate to hypoxia. Okay, otherwise the inner part of the fruit would die. Of course, it can be problematic for post-harvest storage. You know that to do post-harvest treatment to fruits to extend their shelf life, you can lower the oxygen concentration. Can be problematic because it might develop off flavor. You may sense it by yourself. You buy an apple, it tastes, or a pear, it tastes a little bit alcoholic taste because it's actual alcohol. Okay, and interestingly, it is probably at the base of evolution of alcoholism. So I'm moving back and forth from plants to animals, and because I found that this is not our work, okay? So it's but I found this interesting, and I thought it was nice to tell you. So, how did alcoholism evolve? Okay, we are all enjoying drinking a beer or some wine. Okay, why is that, and why can we enjoy so much? So, without going to alcoholism, okay? But why do we enjoy? Why can we enjoy a good glass of wine? It's because we are metabolically fit for metabolizing a reasonable amount of alcohol. So alcoholism and alcohol consumption developed, so alcohol consumption eventually leads to alcoholism, and the idea is that alcohol consumption, there are two theories. One is intentional fermentation of food around 9,000 years ago. So at a certain point, human beings decided, by accident probably, that it was fun to eat something which was fermented, and they started producing fermented food. Or there is an alternative model, which is the primates, so monkeys, might have ingested ethanol via frugivory as early as 80 million years ago. So it's very different. Okay, so two different hypotheses. One is relatively yesterday, but this is a lot, okay? And what is in favor of this theory is studying different monkeys which have a different lifestyle. So this is an orangutan, this is a gorilla. It might look similar. Okay, you would say, they are monkeys. Okay, where was different? Well, yeah. Orangutans spend nearly all the entire life on trees, okay? They live on trees. Orangutans primarily on the ground, okay? If you have a problem with alcohol, which one of the two would you choose to be? Gorilla, yeah, of course. Okay. It's not a nice idea to live on trees and drink alcohol, okay? And this is actually the truth, okay? So, if we look at the alcohol dehydrogenase gene, so alcohol dehydrogenase is the enzyme we have in the liver, which allows us to metabolize alcohol. If we have an efficient alcohol dehydrogenase enzyme, it will be fast. If we have an inefficient alcohol dehydrogenase enzyme, it will be slower, okay? Our friend Akazawa sensei was Japanese, and in Asia, the alcohol dehydrogenase they have is less efficient than ours, okay? So they tend to get intoxicated by alcohol easier, okay? They don't live on trees, so it's not an issue. So if we look at the alcohol dehydrogenase 4 gene, there are two forms. One which is in the gibbon, which is another monkey which lives on trees, orangutans, and then including humans, bonobo, chimpanzee, gorilla, which have an alcohol dehydrogenase version which is active against ethanol. So all these monkeys, including ourselves, can metabolize alcohol, and therefore they can enjoy eating fruits which were on the ground, somehow fermented, because you might imagine that, okay, they start fermenting, and they are enjoying it because it's an alternative source of food. So, of course, the idea was not to get intoxicated by alcohol, but it was to have more choice in terms of food, okay? While, of course, if you were meant to live on a tree, you might imagine that by chance one orangutan got a mutation making its alcohol dehydrogenase very efficient, and he started to enjoying eating fermented fruits, but then it dropped on the soil, and that was gone. Okay, so no options for fixing the trait for evolution, while monkeys living on the ground had the opportunity to develop this additional chance of having fruits even fermented as a food. And which are the concentration of alcohol which can occur in fruits, which are fruits which are eaten by those monkeys, of course? Well, it's a lot, because between ripe and overripe, it ranges between 150 millimolar. This is around 1%, okay? And 10%. Okay, 10% is more than a beer, okay? So, and the Km for alcohol dehydrogenase in orangutan is 1 molar. So it's very inefficient enzyme, while the Km or alcohol dehydrogenase in us, as well as gorillas, is 43 millimolar. So it's pretty efficient. Okay, so this is a work which was published in PNAS, so it's a good journal, and proposing that ethanol, fruit ripening, and the historical origin of human alcoholism in primate frugivory. So it's telling us that, and I am tempted to speculate on a case of coevolution, because of course animals eating fruits then enhance the probability of having seeds dispersed in the environment. You know how I mean. So it is still possible that having some alcohol inside, which might have become a preference by primates, might have induced the evolution to fix some degrees of alcohol inside. It's a trade-off, okay? Let's make big fruits, but then having problems in dispersing. Bigger fruits move slower. But big fruit means also ethanol inside. Well, ethanol inside might be an advantage if then some gorillas will choose to pick the fermented fruits because they like a little bit of having fun, and then they move. So I'm speculating. But it might be the case, because otherwise, why would evolution fix fruit as big as eventually leading to a problem, metabolic problem inside because of hypoxia? So let's move to another aspect of hypoxia, which is developmental hypoxia. So hypoxia in fruits is just one example of hypoxia outside the environment. So it's not due to rain, but it's due to the shape of the fruit. And let's go to the developmental hypoxia, which is another interesting aspect, which was developed again by Francesco Licaus in my lab. And this is an oxygen probe. So we can have you can buy an instrument that actually measures oxygen inside the tissue. It's quite delicate because it's very thin glass needle, very thin, expensive glass needle, which you can push inside the tissue and measure oxygen level. So if we do it inside the meristem of an Arabidopsis, this is what the sensor will measure. So oxygen outside is 20%, 21%. As we go inside into the meristem, oxygen concentration drops to around 1-2%. And then when we push through, so we go down, it resumes. So it appears that there is an hypoxic niche in the shoot apical meristem. And actually, this is the case. So we know, again, cutting a long story short, there is an hypoxic niche in the shoot apical meristem, and there is one protein which is specifically expressed in the shoot apical meristem, which has an MC at the N-terminus. So again, this suggests that there is one protein which is very important for keeping a stem cell being a stem cell, which is oxygen sensitive. So it will be stable only if there is no oxygen. So by doing this, you ensure that that protein is only present in the exact cells which are hypoxic in the shoot apical meristem and not in the other cells. So the idea here is that hypoxia is acting as a positional clue for defining where the stem cells should be and in order to ensure that proteins required for stem cells are expressed there and not elsewhere. And here we can see where this protein is expressed. It's clearly expressed. This is gas, but gas does not have a resolution, but this is a top view, and this is a side view of the shoot apical meristem, and you can clearly see that here we have the expression of the gene. Here we are using a reporter, so it's not. Also, the promoter is ensuring the expression is in the correct site, but it's probably not enough. So you need the gene to be expressed in that site, and to ensure that only if there is hypoxia, then the protein is stable. And this is what happens when you add the actual protein followed by gas. You see that if you keep the plants at 80% oxygen, it's much less stained, while if you keep the plants at 2% oxygen, indicating that the protein actually gets stabilized due to oxygen in the shoot apical meristem. But more interestingly, what happens if we, instead of having an MC protein, we have an MA protein? So we introduce, under the control of the native promoter of a gene, an alternative version of a protein which is no more oxygen unstable. So this version, you just need to replace the C with an A, and the protein gets stable. And you can see that the shoot apical meristem starts proliferating. So if you get rid of the oxygen, of the control by oxygen presence absence, then the protein is expressed ectopically, despite of the fact that we are using a native promoter. And this is enough for leading to an uncontrolled proliferation of the shoot apical meristem, indicating that having hypoxia in the shoot apical meristem is required to ensure the correct development of a plant. Moving ahead, cyclic hypoxia. This is a tumor, I would say in a rat, and tumors are hypoxic, okay, in animals. And there are two kinds of hypoxia in tumors in animals. There is chronic hypoxia, so the tissue is hypoxic here, and hours later is still hypoxic. And there is cyclic hypoxia, so the tumor is hypoxic in this area, but is no more hypoxic later on. So chronic hypoxic, always hypoxia. Cyclic hypoxia, you get fluctuations in the hypoxic level. And this is depicted like that. So the chronic hypoxia is constitutive, while the cyclic hypoxia fluctuates. So we wondered whether this was the case also in animals. And we did find this, which I need to start. Let me see. Yeah, it starts. Okay, so we are looking at the luciferase of an hypoxic gene, and we can see that the hypoxic gene is expressed in the young leaves, but is not expressed now, but then it's expressed. Then it goes down, etc., etc., etc. So this is telling us that hypoxic genes are not expressed in young leaves during the day, but they are expressed in young leaves during the night. And this occurs every day in the life of the plant. And this is cyclic hypoxia in plants, in Arabidopsis. And if we quantify this, so you can of course quantify the luciferase, you see that it's very nice. It goes every night. It goes up, then it goes down, then it goes up, and it goes down. Is really very cyclic, much better than the cyclic hypoxia in tumors. And this is because of stabilization of the ERF. So in young leaves, you make a Western blot. In the ERFs, and you measure one ERF7, you see that it is expressed at a low level during the day in young leaves, but it gets strongly stabilized during the night. So clearly there is hypoxia in young leaves. Every night, young leaves, adult leaves no. But young leaves get hypoxic. So the question now is, Is this just a weird description, or it is useful for the plant? Well, obviously it's useful. The plant wouldn't do it if it was not useful. So it's clearly cyclic epoxy requires the oxygen sensing machinery. If you do it an experiment with a wild type, you see the peak at night. But the oxygen sensing, we have an oxygen sensing defective mutant. It's a pentuple mutant. I said you that there are 5 genes encoding for RF proteins in Arabidopsis. We have a mutant which does not have any of the 5, so it's very useful to study oxygen sensing, obviously. And you can see that here that the oxygen sensing defective mutant is not fluctuating, so it's not an alternative signal. It's that one which is working. And is cyclic epoxy relevant for plant growth? Well, we measure the metabolism inside the cells at night, obviously young leaves, at night and during the day. And we used a technique which is exploiting NADH autofluorescence. If you go to the confocal and you do this technique, you will get this pseudocolor imaging, which is indicating you that there is a lot of NADH in these cells, while when you have this signal, you have very little NADH. A lot of NADH indicates anaerobic metabolism, okay? If it is aerobic, all the NADH will be used by the mitochondria. Okay, if NADH accumulates, it means that the mitochondria are not working nicely. Okay, so clearly there is a change in metabolism. So the metabolism is hypoxic, as a consequence slower, because of course there is less chances of utilizing sugars, and faster during the day in aerobic metabolism. But so this is the situation during the night, hypoxic metabolism, during the day, aerobic metabolism. So it's not just that the genes are fluctuating. The genes are fluctuating because these leaves get hypoxic during the night. There is no photosynthesis at night, so it's more chances of getting hypoxic. And the actual metabolism is hypoxic. So it's not just gene regulation, but the actual metabolism is hypoxic. If you use an oxygen-sensing mutant during the night, the metabolism is aerobic. So it's unable to sense hypoxia, and therefore there is no switch between aerobic. So this implies that the amount of oxygen which is present would be enough for aerobic metabolism, but there is a decision taken to switch the metabolism to hypoxic during the night, to slow down metabolism at night, and resume fast metabolism during the day. So is a signal which allows the plant to understand whether it is night or not, and it is important to slow down metabolism at night. Why this? Because of starch. Starch is degraded during the night in a way that it is slowly degraded so that the starch which was accumulated at the end of the day will last for the whole night until light comes back and the plants can resume photosynthesis, etc., etc. But look at the amount of starch. This is the beginning of the night, this is middle of the night, etc., etc. And look at the oxygen sensitive defective mutant. After a few hours in the night, it already used all starch. So it will enter sugar starvation, and it will eventually have problems, okay, of keeping the metabolic machinery active. So this is what happens in an Arabidopsis plant. During the day, in the beginning of the day, there is no starch. Then starch keeps accumulating. We have growth of a plant. Then during the night, there is no more light, and starch which has been accumulated will be utilized until you reach the next day, and the cycle of starch synthesis degradation will start over. So this is the ideal situation. During the night, you have a linear rate of starch degradation, which is actually what happens, okay? We have a linear rate of oxygen consumption until the end of the night is reached, and then we have a linear rate of starch synthesis during the next day. And this what happens in the oxygen-sensing mutant is that. So the starch degradation rate is no more linear. It is too fast at the beginning of the night. You get starvation at night, and this has an impact on the overall performances of a plant. And you can see here, when we measure growth of a wild type of the mutant on all 5 ERF proteins, so this is the oxygen-sensing mutant, And the growth in the beginning, the plants are the same size, but as they start growing under a long day cycle, long days means short nights, okay? So it's even more tricky. And you see that the mutants start slowing down growth, and after a few days, you start having the plants were same size in the beginning, and this plant is smaller than this plant after a few days. So having the ability to sense oxygen in a fully aerobic plant is important because measuring oxygen level is important to set the growth level to the optimal rate, so that there is enough starch left for ensuring growth by the plant. So, getting to the conclusion, so we demonstrated we have hypoxic niches in aerobic plants which generate hypoxia. Hypoxia will inhibit the PCO, which is the enzyme which oxidizes the MC protein. The MC protein you might have ZPR2, which is the one I showed you for the shoot apical mechanism. There is another one which is the VERN2, vernalization 2. It's an important protein for flowering as well. And these will impact on plant development. You have submergence, and in submergence you have hypoxia. You have the same mechanism, so we have another ERF protein which is responsible for the stress responses. So the key mechanism is having an MC protein which is translating the presence of oxygen into a phenotype, which can be shoot development, it can be stress responses, etc., etc. And then there is rice, which, weirdly enough, does not utilize this mechanism. And I'll try to convince you that there is a good reason for not doing that. Rice exploits ethylene, which induces sub-1A, which is an MC protein. But this is an MC protein which is different because the MC is protected by folding of a protein with a C-terminus that folds to protect the C-terminus. So evolution made the mechanism of tolerance of rice to submergence independent of the MC mechanism, because having a mechanism which is relying on MC would have impacted many other aspects of the growth of a plant. So the plant needs to be able to distinguish between submergence and hypoxia. Submergence sensing is via ethylene. It's a nice, simple mechanism. The plant is submerged, ethylene gets entrapped. So ethylene entrapment is the mechanism by which plant sense submergence. And submergence is through a mechanism which is ethylene dependent but oxygen independent. While hypoxia and all the oxygen sensing mechanisms very reasonably was developed mostly because of hypoxia as a developmental signal rather than as a stress signal. And this led to a number of phenotypes which are dependent upon hypoxic niches in otherwise aerobic plants. Dozo. Ides? Ides. So that's it. I will be glad to take your questions, if any. Thank you very much for your attention.
Bueno, Pito Pito Gorgorito, Karen, que está la primera por aquí.
Hi, great talk. I really enjoyed it. I have one question. You said that the mechanism is shared between mammals, humans, and plants. What happens in yeast or other
no, we don't think it would happen in yeast. I think that the endogram pathway is not that well conserved in yeast. So as far as we know, it is not conserved in microbes.
Well, the question is, what about seagrass, Zostera?
Well, seagrass are hardly hypoxic. It's a good point. I always wonder why seagrass are not, whether they are actually. So they, thanks to photosynthesis, they produce a huge amount of oxygen. So seagrasses, and seagrasses has an anatomy which is oversimplified. So the tissue is simpler than land plants. And therefore, they should not be affected by hypoxia. However, I must confess that apart from works done in Chlamydomonas, which is a single cell algae, in which we demonstrated, because we actually did the work, we demonstrated that there is hypoxia. So and the mechanism is similar. I don't know about the sensing mechanism, but the mechanism involves ethanol production, alcohol dehydrogenase. So it's not that different from what happens in plants. I must confess that there is not many people working on algae which are interested in studying this. So I wouldn't exclude the possibility that algae do have their own mechanism for oxygen sensing and adaptation.
Because one simple thing would be to see how those genes important are conserved in
Yeah,
sequence,
it's
right?
exactly what we did with Chlamydomonas, and we found that for alcohol dehydrogenase they conserved. There is a work which recently came out, again it's by Francesco Licausi, which was from my lab, now he's in Oxford, and about the evolution of the oxygen sensing mechanism. And what it came out is that the oxygen sensing mechanism that I described was evolved since the colonization of land by plants. So it is not present in algae.
Yeah, but this is a plant that came back to the sea. I mean, so it's
well,
they were terrestrial plants
if you
that
talk
came-
about plants like Poseidonia, I would agree, but it's not an algae. Okay, so yes, in terms of Poseidonia, I'm sure it has the mechanism in place. I'm sure. I never checked, but I'm pretty sure that it's there, but not in algae.
Great talk, Pierdomenico. So I had like a couple of very naive questions, because the first one is because I am roughly having problem with alcohol, so it's
Okay,
something that
well,
I'm
it's not
very
a problem.
I mean, the alcohol has a problem with me now.
Okay, okay, that's better.
So regarding all the part that you talk about apes and,
Yes.
and alcohol and stuff like that. So what about other animals that eat the fruits, like
I think they do have the alcohol dehydrogenase, which is the good
what
one.
about insects? Because I mean,
I
like
don't know.
For no, because the thing is, like, for a gorilla.
Well,
so
for
drinking a
example,
beer a gorilla is nothing.
Drosophila, I would say, but I'm really speculating a lot. I would say that a Drosophila would be adapted. They are fruit flies. They would forage on fruits, and if they are rotten food, they even like them more. So I would say yes, but I'm really speculating on that.
Because those should be super active, and we can get them. It would be very useful.
So your question is an interested question. So you want to find a way to...
So and another one regarding hypoxia. So I have seen it in my garden. So I forgot like the watering system.
Yes.
Open it overnight,
Yeah.
and then the following day it was like floating
Yes,
in the garden.
okay.
But the leaves were completely fine, but
Yeah.
the plants die. So is the roots also
Yes,
having like a lot of
yes,
stress?
yes. Clearly, the roots are the mostly affected. So we have to distinguish flooding from waterlogging. So what happened in your garden is waterlogging. So it's soil saturated with water, which is by far enough to kill the plant, because inevitably, unless of course the plant is a plant which is well adapted, so with an parenchyma system in which there are bigger spaces connecting the leaf to the root system. Okay, you might, otherwise you wouldn't be able to explain how plants live in a pond. Okay, these plants have no problem, but hypoxia has nothing to do with these plants, because simply they are avoiding the stress through a air transport system from the upper part of the plant to the underground part of the plant. But your lawn was not adapted.
Okay. No, but something that you should keep in mind, and this is a project for the future for you.
Your lawn.
So,
Or the alcohol.
my garden.
Or the,
So-
or the alcohol.
So you know, like we have like all these lantanas plants. All
Ah,
the,
okay.
all the
I know
colors,
lantana, yeah.
all the colors of lantana, and the only ones that die were the one that has the Spanish flag colors. The rest were totally fine.
Okay, okay. So it's, you see, biodiversity in action.
Yeah, exactly. So you can check that.
Well, actually, I must say that I couldn't tell you everything, but there are huge differences in tolerance in Arabidopsis. You know that thanks to the 1,000 Arabidopsis genomes project, we have 1,000 different ecotypes of Arabidopsis being sequenced and geo-referenced. So we made a landscape genomics approach by choosing sites, by crossing the geolocalization of different Arabidopsis ecotypes with climatic data. So we have Arabidopsis ecotypes which are typical of areas where it rains a lot, and other ecotypes which are typical of areas where it does not rain a lot. And by doing a GWAS approach, an EGWAS approach, environmental GWAS approach, so merging the climatic data, the genome sequences data, we identified one gene which is a positive regulator of ERF7, which is present with the SNPs, which are only present in the ecotypes of Arabidopsis growing in wet regions. So, yes, within the very same species, you can have adaptations at the genomic level which are due to the climate in which they... So we can't say Arabidopsis is surviving 2 days, which Columbia is surviving 2 days. But you can have ecotypes of Arabidopsis which are identical to Columbia, which simply because they were they grow typically in a wet area, they can last 1 week under submergence.
Well, thank you very much. It was very interesting, and the question of Pavel were even more interesting also. But I have a very much simple question. When you show the mechanism by which the plant is able to recover once you remove it, and you put back the oxygen.
Yes.
And you show that that's basically because it's sensing the oxygen through the degradation.
Yes.
I was okay. But then I have a question, because in the picture that you show at the beginning, you show that the protein was in the plasma membrane, which has there are oxygen there. But the protein is there, and then moved to the nucleus.
Yes.
My question is, how is possible that the protein is stable in the plasma membrane when in condition with there is oxygen, and how, or is known how it's moved to from the plasma membrane to nucleus?
The protein is stable because the MC, which is the sensitive domain, is docked to another protein, which is ACBP, which is a plasma membrane protein, ACBP1, and is docked there. So, originally we thought it is there because you need to have a fast response, so you don't have time to wait for a newly synthesized protein to be stabilized. You want some protein to be ready to go nel nucleus. Now I'm a little bit more skeptical about that. So I would say that newly synthesized proteins during the initial hypoxia would be enough to trigger the response. However, we have to say that the response, the transcriptional response to hypoxia occurs within 5-10 minutes of hypoxia. So if you place a plant under hypoxia, the response is that fast. So it still would be compatible with having a reservoir of protein stabilized in the plasma membrane. The undocking is due to changes in the... So the protein has a ACBP protein, which is binding fatty acids, okay, originally. Okay, so it was proven that during hypoxia there is a change in the composition of the fatty acids in the plasma membrane, and this might lead to the release. Okay, so it's the interaction of a CBP protein with fatty acids in the plasma membrane, which if the plasma membrane fatty acid composition change, then the protein releases the transcription factor. So it's a complicated mechanism. So again, I think it's very nice and sophisticated. I do not know whether the relative contribution of newly synthesized proteins versus the stored one is that relevant.
Okay. Thank you for your talk. I have a curiosity about the different type of variety of rice.
Yes.
So how they develop, like nature selection or human breeding?
No, it's not. It's mostly natural selection. The SAB1A gene, for example, was found originally in land races of rice which are not very productive. Okay, so now we have a scuba rice because the trait which was found in unproductive rice varieties was introgressed in highly productive varieties. Okay, and so it's very different. Of course, the deep water rice is typical of regions where it rains a lot. The water level raises 3 meters and it does not get back, because otherwise the plants would. it's a floating rice. It's called a floating rice. It should float. It's harvested with boats. Actually, it's harvested with boats. It's a niche of kind of wild rice which is exploited by agriculture. While the sub one is evolved through similarly to ecological niches where it rains a lot, and now it is introgressed into the. By the way, rice has VRF7 genes which are oxygen sensitive, but they are not the one linked to tolerance. So I don't want to convey the message that rice is totally different. No, the oxygen sensing mechanism is there, but it's not exploited to be tolerant. Another mechanism which uses the very same genes, but with a weird modification in their oxygen sensitivity.
Okay. And I have a second one. It's about the methionine that is removed.
Yes.
You say it's removed, but how is that?
There is an enzyme which is
So it's also,
MAP.
but it's also oxygen.
No, it is not. It is not. It is
so
a reaction.
It's directly a
it's spontaneous, enzymatically driven, but spontaneous reaction in which we have a work in progress which might say something about oxygen regulation of MAP as well, but it's not yet done. So.
More questions?
Thank you for your talk. I have a question, maybe it's a naive one, but the flooding and all this condition is like a hypoxia condition outside of the plant, no? But then in the night, the plant is generating one hypoxia condition inside the plant, no? Because the percentage of oxygen is the same by the night and the day, no? So my question is, how the plant is generating this hypoxia condition inside of itself?
Okay. So there are two hypotheses. One is thickness of a tissue and eventually waxes, preventing from the outside to the inside the influx of oxygen. The second one is the metabolic rate. If the metabolic rate inside the tissue is faster than diffusion of oxygen from outside, you generate hypoxia. Inevitably, you also have to consider oxygen generation by photosynthesis, which is from the inside. So in the end, this explains why cyclic hypoxia is during the night, because there is no contribution of oxygen produced by photosynthesis. And while in the shoot apical meristem, there is an ongoing work by Dan Weitz, so I don't want to spoil his work. But the hypothesis is the one I said. On one side, diffusion rate outside to inside and the rate of consumption inside. Strongly active tissues, young leaves, are more likely to have hypoxia endogenously because the rate of respiration is so fast that it might be stronger than the rate of influx of oxygen.
More questions? Yeah.
Thank you for the talk. I have a question. Does it exist in nature any other thing that causes hypoxia in plants apart from the ones that you showed to us? I mean, fungi, floods, other diseases, nights and days, alternatives, apart from this one. Thank you.
Okay, so about pathogens, necrotrophic fungi apparently do. Biotrophic fungi might not do. So it's still unclear. We never explored insects, for example, or viruses infection, so it's a possibility. Anything which would induce a burst in respiration in the plant might lead to hypoxia. Okay, then it's complicated to understand whether it is an necrotrophic fungi. The fungi induces hypoxia. Hypoxia kills the cells, and the fungi gets an advantage. Or hypoxia is induced at the site of infection. The plant is able to counteract hypoxia and is able to fight back. So the trade-off between the fungal attack and the response of the plant is something that has to be defined. We are working on it. It's very interesting because, of course, plants which are typical of wet areas are also more likely to be prone to fungi, for example, because high air humidity is another not studied trait And then there is chemical induction of hypoxia. We selected a number of molecules which act as the inhibitors of PCO, and therefore, by inhibiting the enzyme which oxidize the VRF, they can induce hypoxia in air. And these are eventually interesting molecules to prime crop plants for hypoxia. You spray the chemical, you induce a pseudo-hypoxic response in air, and the plant is primed for, so maybe for your garden, I should provide you with, so you watch the meteo and you are ready to spray with a chemical. No, the idea is exactly that, so to have a possibility to prime plants with a chemical. So it's an evolution towards a possible chemical intervention for preventing damages by hypoxia.
More questions? Well, I have one. In fact, there are two. I'm wondering to which extent the response of plants to hypoxia or submergence are mediated by relationship with microorganisms, because all these things you have done not in sterilized conditions. I mean,
yeah, of course. Well, some of them were done in sterile conditions.
Yeah, because you are also working on, you said, on exogenous RNA signaling and so on microbes emit RNA and so on. So, and many other molecules, of course, hormones and etc. etc. So, do you have any idea?
So the mechanism works in under sterile conditions as well, because of course you can grow Arabidopsis under sterile conditions. You might still have endophytes, but who knows? For sure, hypoxia is influencing the microbiome. Of course. That is influencing a lot. And a colleague of mine in Pisa is working on the rice endophytes, which might be contributing to rice tolerance, because of course if the microbial community is also influenced by hypoxia, it might be selected with endophytes in rice, which might be vertically transmitted as well, and therefore there is all this part of the plant microbe interaction under hypoxia. But in terms of oxygen sensing, I would say no, it's not influenced, apart from microbes causing agrobacterium infecting a plant is causing hypoxia.
Yeah, well, but also emitters of ethylene, according to your.
Oh yeah.
So many.
Yeah. But that would be. So there it opens another field, which is: are there any alternative sensing mechanism besides the one we described? I would say yes, and ethylene would be very interesting to be studied further, because what makes the difference between a submergence and endogenous hypoxia is the presence of ethylene. So it's a nice mechanism to discriminate between the two by a plant.
Yeah. Okay.
One last question. Going back to the fact that the sensor is in the plasma membrane and they move back, and relying on the pre-existing protein or synthesizing new ones, maybe. I mean, for active translation, you need oxygen and an active metabolism. So might be better to have the protein already there in order not to rely on translation, which actually drops very much under hypoxia?
You have translation under hypoxia.
Yeah, but
it's selective. It is selective. And these genes are selectively translated, so they are loaded onto polysomes. And yes.
But I mean, you are again relying on a mechanism that in general drops, so maybe it's like the combination, or maybe it's a good idea to have it already prepared just in case?
That would be our original idea, and it's an idea that after more than 10 years holds. As I said, I still think what was puzzling in the beginning was because there are 5 ERF genes. 2 of them are strongly hypoxia inducible, and initially we focused on these 2. They were the wrong ones. If you want to it happens. If because you are misleaded, okay? You find which is the one which I'm studying hypoxia. There are 5. 2 are induced by hypoxia. These are the interesting one, okay? But they are not, because you do not want. If they are induced by hypoxia, they are downstream of sensing. They are themselves. And the 3 out of the 5, free are those important, and they are constitutively expressed.
Yeah, that was the point. Like, if you wait for the induced translation
Yeah,
after
yeah, yeah.
hypoxia,
There is a lag.
You're missing the sensor.
Yeah, yeah.
So
Well, you have to imagine these promoters are very strong. So that was puzzling. I mean, again, you try to see, are these important? Let's make an overexpressor. Not much of a difference. Because why? You overexpress an MC protein. There is a lot of messenger RNA, but then all the protein is degraded. So we couldn't see, we didn't have yet an antibody. So in the beginning, you don't have all the tools. Yes, on one side you have a gene, three genes actually, which are very strongly transcriptionally activated constitutively, always. And then you have the problem of how fast it is inhibition of enzyme which oxidize the protein. So theoretically, the amount of protein which is translated is a lot, but it is so rapidly degraded that there is no time to catch it with a Western blot, okay, because there is nothing. Apparently, when there is no oxygen, it is stabilized within minutes. Is this protein all coming from a membrane, or is it coming from newly synthesized protein? I would say both. I cannot exclude that during the first 5 minutes, there is a crucial contribution of the protein which was stored in the membrane. The problem is that there were attempts with the mutants of ACBP, so the docking protein. So, but these mutants are lethal because this protein has do other stuff. So it's not that easy to get rid of a docking system to have the proteins only active if it is newly synthesized, newly translated, as hypoxia is established.